the nucleosome is going to be first level of organization, which is also known as Berian string model, which is going to have a histone protein in the form of octamer as a core protein, where the DNA is going to be wound around it. It can form a core DNA and the linker DNA. It is the first level of organization. A second level of organization, a first level of organization can form about 7 nm fiber. The second level of organization, it can form a 30 nm fiber. Therefore, a second level of organization consists of 6 nucleosomes. 6 nucleosomes are going to be binded off by H1 protein, which is known as a linker protein. A linker protein comes into the act in the second level of organization. So, the intranucleosomal contact is going to be taken place between the 6 nucleosomes by H1 protein. It is also positively charged, and negatively charged DNA is going to be contacted, which means a linker DNA is attached by the linker H1 protein by intranucleosomal contact. So, here we can say that H1 protein is going to make it to tighten the DNA further. Thus, the nucleosome can be disintegrated by micrococal nuclease like enzyme, but a second level of organization by H1 protein makes it into nucleosomal contact and makes it to tighten the DNA. Here we can say that there is a nucleosome. So, here the H1 protein gets attached and makes it to tighten. Here it is a nucleosome. Like this, a 6 nucleosome must be there. The 6 nucleosomes can makes it to tighten like this. It has more intervals of about 7 to 10 nm fiber. But when there is a nucleosome, it comes to 30 nm fiber. A second level of organization has 2 types of organization. When there is a tightening, the tightening of H1 protein can also be made by 2 types. So, as we said that there is a linker DNA which is going to consist of 146 base pair, 114 base pairs. A core DNA is going to consist of 146 base pair. So, a linker DNA is going to be associated with H1 protein with the core DNA of 146 base pair. This makes it to tighten. So, the two distinct regions are going to be associated, core DNA and linker DNA, by H1 protein. For the 6 nucleosome, it made that. So, it's going to be tightened, but the way of tightening can be made in 2 ways, by solenoid model and zigzag model. First, we see it individually. In the solenoid model, there are about 6 nucleosomes per turn is going to be made. But in the solenoid model, the turn is going to be arranged in the helix form. That means a super helix form is going to be made. So, both is going to form a 30 nm fiber, but the helical pitch is going to be 11 nm. Because it is going to form a super helix, the pitch is going to be 11 nm in the solenoid model. Which means a nucleosomes are stacked around in the helical manner in the adjacent sites. Stacked in the helical manner. That means a nucleosome upper side of phase core DNA is going to be adjacent to each other, and a linker DNA is going to be buried in the center. That is a linker DNA in the center. So, this solenoid model itself can be made as 2 types, a single linear array or double row array. In general, a helix model can be single linear or the double helical. Like this, a nucleosome can be arranged in double manner or a single linear. But a single start is going to be a most accepted model and mostly prevalent model. A solenoid can pack a 40 nucleosome to chromosomes. Here we can say that it is going to be a core histone. This is a core DNA. This is going to be a linker DNA between two nucleosomes. It is a double row array of solenoid model because there is double row. This is going to be adjacently packed. This adjacent packing of the nucleosomes is going to be known as solenoid model. Where here we can see that a linker DNA is going to be as the middle is going to be a linker DNA, and in the adjacent form there is a core DNA to be present. This is the solenoid model where a helical like structure is formed. Here we can say that helical like structure is formed. Next, a zigzag model. Zigzag model is going to make a zigzag like position arrangement of nucleosome. So the zigzag pattern of arrangement is going to be made. It also has a 40 nucleosome. 40 nucleosomes can make a chromosome in the zigzag model. Both are going to make a 30 nm fiber and 40 nucleosome, and each 30 nm fiber consists of 6 nucleosomes. 6 nucleosomes are going to be packed off. 6 nucleosomes are going to be packed off, and it can pack about 40 nucleosomes in the chromosome. Is done by zigzag model. It too has a linker DNA at the middle. So here we can say that linker DNA, but it is a loose. It has more interval than the solenoid model. Solenoid model is going to be a more tightened form than the zigzag model. So here we can see that zigzag position. A linker DNA is going to be have more interval than the solenoid model. Here we can see the solenoid model. In the linker is going to be tightened, but in the zigzag it is going to be more loosed.